Good. Okay. Well, good afternoon, everybody. So I'm a professor in the computer science department, and I lead the genomics and bioinformatics cluster initiative here at UCF. I work in the area of computational biology. That means I develop algorithms basically for analyzing large genomics and other omics data sets, and using our methods to really tease out the underlying biology of the biological systems that we study. Previously in my career, prior to joining UCF in 2016, I've also worked in nonprofits and in the industry as well. I've been involved in several seminal genome sequencing projects, including the Human Genome Project and the Mouse Genome Project. What I wanted to talk today really are about microbes, so the smaller organisms. So microbes are invisible to the naked eye, but we have been, scientists have been able to study microbes and observe them for centuries now. So in fact, since the 17th century, and the invention of the microscope and the work of Robert Hooke and Antony von Leeuwenhoek and all of those folks. But really, I think it's fair enough to say that our understanding of microbial communities, the diversity of microbial life, and the molecular basis about microbial life, all of those progress in all of these areas have really taken off only in the last 2 to 3 decades or so. And this has been made feasible mainly through the adoption or mainly due to the availability of DNA sequencing technologies and the use of cultivation-independent approaches to study microbes. So in fact, I've actually been involved in several large-scale genome projects to study microbial communities in different environments, including in the open oceans, in the human environment, airborne environment, and soil and several other environments. And what these studies, not just work from our group, but work from the overall research community has revealed is the incredible diversity of microbes that exists on Earth. And in fact, the estimate is that these invisible organisms to the naked eye, these organisms make up roughly half of the biomass on Earth. And if you were to construct an evolutionary tree of life that links all of the organisms, all of life on Earth, you will find that in fact, almost all of the diversity in Earth is explained using these tiny life forms. Us as humans and eukaryotes, we occupy a tiny leaf in this big tree of life, if you will. And what's interesting is that these technologies is now, have resulted in enough newer discoveries that the tree of life keeps getting updated very so often. So it's an interesting situation that we live in. In fact, in the human body itself, there are as many microbial cells that are in contact with our body, in our body and on our body as there are human cells. So pretty much, half of us is really not us. And that leads to some interesting philosophical questions as well. But for our purpose, we're going to be focusing today on the gut microbiome. And the gut microbiome actually plays an incredibly important role in human health. In fact, because microbes have been around for a lot longer than we have, they've actually co-evolved with us. And therefore, it's not a surprise that their composition, structure, and the type of microbes actually do play an important role in influencing human health. In fact, microbes produce for us certain nutrients and vitamins that our human body cannot produce. They prime our immune system. They have a lot of different functions. And when the community structure of a healthy human or a healthy microbiome changes, that results in what's referred to as dysbiosis, and that can lead to a whole bunch of different diseases and imbalances. Including metabolic disorders, liver disease, cancers, and a lot of recent work has gone into trying to understand microbial role in disease and these microbial biomarkers, especially in the context of diagnostic and therapeutic purposes. So today what I wanted to do was, so before I get to the problem for today, and so the way microbes have been studied most recently has been using genomic-based approaches, and in particular what's referred to as metagenomics. So this precludes the or avoids the necessity to cultivate organisms. You can basically take a sample from any environment of interest, extract the DNA, sequence it, and generate DNA sequence data. And then it becomes a really interesting computational challenge to figure out what microbes are actually present in these communities, i.e. trying to assemble the genomes of these organisms and trying to infer, what properties these guys have and what functions and what roles these have. And so this has resulted in a whole slew of really interesting types of computational problems in this field, many of which my groups have worked on in the past and continue to work on to date as well. But today I wanted to focus on a particular situation when that arises when you study microbial communities, namely this problem of inferring microbial associations. So we have to keep in mind that in a microbial community, it's not just a single type of microbe, but you have microbes from different taxonomic, different parts of the taxonomy, different types of microbes, different metabolic capabilities, they interact with each other. And it's really these interactions that determine the community structure and function, and also determines the abundances of these constituent microbes in the communities. And so trying to understand these associations is an incredibly important component, because we can use this information to figure out who are the important players in the community. And the one way to do this is to kind of turn this problem on its head. So we can actually use sequence data in which we've generated what's referred to as a sample taxon matrix, where what we've done is gone to an environment of interest and collected samples, sequenced it, and then figured out how many counts of each of the different taxa are there. And then we can use that information to try to infer or figure out this graph referred to as an association or an interaction graph, in which the nodes represent taxa and the edges represent or denote the strength of association. So it could be positive associations, negative associations, and so on. Of course, frame this problem very simply, but it's a little bit more challenging than I've noted here, including the fact that sequence data are compositional in nature, and therefore you can't really directly apply correlation methods. One has to be very careful and really also keep in mind that often what happens is the number of samples is often not high enough to accurately estimate the various edges in this particular graph. And so there have been a few different methods that have been proposed, including latent variable modeling approaches, and another approach that involves basically taking the count data, log transforming these data, and then assuming that the underlying data were produced using a Gaussian model, and then inferring the covariance matrix, the precision matrix of this underlying Gaussian model. So these are what are referred to as the Gaussian graphical models. Another assumption that's often invoked is the sparsity assumption, which does make sense in a microbial community setting, given the often the physical and chemical barriers that exist, and therefore the fact that not every taxon interacts with every other taxon, right? So the number of edges in this graph is often not quadratic, but linear in the size of the graph. And so the Gaussian graphical models are also a really useful framework, because they actually can be used to capture independence between taxa interactions. Anyway, our contribution to that area involved actually developing more accurate methods that with fewer false positive rates compared to some of the previous existing methods. We used the methods that we developed to study the microbial structure, association structure across healthy gut microbiomes across 4 different populations, namely American, Indian, European, and Japanese populations. So by the way, these datasets were previously published. We took these data, sequence data, and used that to do association studies, right? So I'm not going to go into the details of how we computed the graphs, but just wanted to show you what the results of the graph structures look like at a high level. So by the way, these are, so on the circumference, we have the nodes represented by the different species. They come from various microbial phyla. And just the point to note here is that the graph structure here across the 4 different human populations actually do look quite different. So for example, a lot more edges between genomes from the Bacteroides group in the American network, whereas for Europeans and Indians, you tend to see more of edges between groups from Firmicutes. Anyway, once you zoom into the graph structure, you can start identifying a whole bunch of really interesting properties, including the fact that most of these associations in these communities, microbial communities, tend to be positive. Definitely, it's a case that these graph structures are not random in any perspective, right? We've actually tested for those and found that that's not the case. We also see that most of these positive associations are between groups of bacteria that are actually taxonomically and functionally related. And while negative associations often occur between species that are quite distantly related on the tree of life. This has implications in terms of when you're trying to look at functions of these different communities. We also used certain graph properties to identify hub species, sort of important species within each of these different community structures. One of our other analysis also shows that even though there's a huge amount of taxonomic variation, from a functional perspective, we don't tend to see as much. And that has important implications. It could be the case that you have taxa that maybe from different parts of the tree of life that could have similar functions, could be used as replacements in certain situations, right? We've actually taken a framework and an approach and used it to study microbial community structures in the case of different diseases, in particular, colorectal cancer and inflammatory bowel disease. And a couple of different recent publications. Please take a look at these if you're interested. In these publications, we identified microbial biomarkers that are indicative of the differences in community structure between healthy and diseased communities, and also identities of microbes that could be playing fundamentally important roles in the progression of disease. Okay. In this area of microbial associations, there's also another approach whereby you can try to model the count data that you get from sequencing using various distributions. So one popular distribution that's used is something referred to as a multivariate Poisson log normal distribution. So the idea here is that the observed count data is generated by independent Poissons whose logarithm means are linked together by a hidden Gaussian distribution. So sort of a two-layer distribution, if you will. And this was proposed back in the 80s by Aitchison and others, and it's been used in this context as well. Again, the point here is that our goal would be to estimate the precision matrix associated with the hidden Gaussian distribution. Now, this particular distribution, while it's interesting, it can capture both positive and negative associations, does have challenges. In fact, you can try to, in a likelihood setting, you can solve for its parameters. You can't compute it exactly, the probability density function, but you can actually estimate, you can compute its moments, by the way. We took this framework and extended it to study this problem of inferring from large microbiome data sets multiple microbial networks. So the rationale for this is that you can often have, in case of large data sets, large cohorts, if you will, you may actually be sampling from microbial communities that do not have just one underlying covariant structure, if you will, but multiple covariant structures. So in this case, I've denoted it by K different components. So this, in essence, can be formulated using a mixture model problem. It has some really interesting computational properties, and the way for estimating the parameters is non-trivial. We actually ended up using a principle that was first made popular by Ken Lang and others. We had a publication back in 2019 in ISMB on this. So please take a look at this if you're interested. Most recently, we've actually been looking at the model selection problem associated with this particular framework. And especially using variation approximation algorithms to figure out the number of components associated with this network. So what's the goal of all of this? Well, one of the big picture goals is once you have these methodologies in place, our next step is to take existing large microbiome data sets from different environments and really compute these microbial association networks and really get at this idea of inferring the rules of life or how these microbes interact in different environments, what some of the common features are and what some of the differences are. And we want to link that with the various metadata that are associated with many of these studies. So we hope to then take these methodologies and apply to various environmental sciences, but also to human health studies as well. Some of this work has been funded recently by a big NSF grant that I received. This is, like I said, this is ongoing work. But I would like to conclude by thanking my students, Mark Loftus, Saif Hasune, and Sahar Tavakoli for some of the work, some of the results that I showed you in the earlier slide. So I'll stop now and happy to take any questions. Thank you.